Hello students, welcome to EPG Pathshala. I am Dr. Nirupama Trihenpati from Institute of Liver and Biliary Sciences. Today we are going to talk on the module Biotherapy for Viral Diseases under this paper Molecular Therapeutics. After completing this module, you should be able to understand the viral infections and related mechanisms of inflammation. Objective of this module to make you understand what are the immune pathogenesis in viral infections. Means whenever there is any viral infection, how it changes the immunity of the body and which cells. And so if we will understand first that during viral infection how they change the immunity of system, we can target those cells for the biotherapies. So our third objective will be therapies targeting those cells, whether they are causing the immune suppression or they are causing the immune exhaustion. This figure explains you that whenever viral particles, full virus or viral antigens are seen by the antigen presenting cells, they secrete the interferon alpha and beta, which itself will activate the dendritic cells or the other natural killer cells. Now these dendritic cells and natural killer cells will make the more cytokines, and those cytokines will enhance or induce the adoptive immune system of the body. So once the viral infection or the viral particle is sensed by the cells, they secrete the cytokines, and those cytokines now they induce the adoptive immune responses to clear the virus cells or the Kupffer cells. But there are problem of the other innate immunity cells like monocytes. Monocytes are increased during viral infection and also maintain their activity. However, their interplasticity to the macrophages is disturbed. Whether they become the macrophages or not, which are CD14 and CD68 positive, that is very important. And these other initiators of innate immunity may have tempered viral infections. Problems of innate immunity in viral pathogenesis. There must be the decreased expression of TLRs on viral infection. There could be the defective monocyte macrophages, as I already told you, that inherent plasticity in response to injury or viral infection leading to inflammation. This ratio changes, and how much it changes, it depends upon the capacity of the virus and the deteriorated pathogenesis. Defective NK cells. How they are defective? Because they don't produce the interferon gamma. They have the limited secretion of interferon gamma. So those three cells are innate immune cells, and these have the problems when the viral infection. As I explained you earlier, that it is important to understand the mechanism or the pathogenesis over the viral infections, that which cells are involved, so we can start thinking of boosting up those cells in the innate immune system, or start thinking of creating the biotherapies along these cells. So now this figure shows you that how adoptive immunity works in viral infection. Once the virus is sensed by a cell, it induces the innate immune response and indirectly induce the adoptive immunity. Now in adoptive immunity, there are different kind of T cells. T cells, naive T cells, which they have not yet activated, but once they are activated, they start secreting the cytokines which lead them to the Th1 kind of T cell population, Th2 kind of T cell population, Th1 kind of T cell population is the cytokines, and they differ into the CD4 activated cells, CD8 activated cells, and then they destroy the virus. Now the third population comes regulatory T cells, and that regulatory T cells are the making the homeostasis of immune system, that they balance the CD4 and CD8 because they suppress the unwanted proliferation of these CD4 CD8 cells. In the adoptive immunity, there is the helper T cells also. Those helper T cells, which we call as Th2 helper T cells, they help the B cells to mature and to make the antibodies against the viruses. And now these antibodies, they neutralize the virus and they behave in the antiviral kind of therapy. So if this is the innate and adoptive immune system, it really boost up when the viral infection. However, if the person is not healthy or compromised, then innate and adoptive immunity is compromised. I am explaining, when there is a viral infection and acute viral infection, at the time of viral infection, early immune events, how they vary. In the early immune events, viral specific CD8 T cells are deteriorated, they are influenced. Fast ligand leading to apoptosis, that is disturbed. NK and NK T cell ratio, that is disturbed, which plays an important role. And certainly adaptive immunity induction is important to clear the acute viral infections. If these four parameters are disturbed, viral infection will persist and it will become chronic. In the acute viral infection, all those four parameters are strong and they are able to clear the virus. However, if these four parameters are disturbed and dysfunctional and in low quantities, therefore then the viral infection will persist and become chronic. Other than innate immune and the NK T cells, monocytes, macrophages, there is a mechanism of suppression upon viral infections, and that mechanism of suppression is operated by the regulatory T cells. There are two kinds of regulatory T cells, natural Tregs and the induced Tregs. Natural Tregs comes from the thymus and they are very less in the periphery. It reaches in few quantities or the few numbers in the periphery. Their way of suppression the cells is contact dependent, that they come close to the other cells like NK cells, dendritic cells, other T cells, monocyte, macrophages, and then they suppress via the production of TGF-beta and IL-2, or they destroy the cellular content of the other cells. In this way, they behave. However, whenever any viral infection is there, those viral infection induce these CD4 positive naive T cells and they convert these naive T cells into induced regulatory T cells. How they suppress to the other molecules? They don't come through the only contact dependent, they are even cytokine dependent. These regulatory T cell, induced regulatory T cell, they secrete the IL-10 TGF-beta, and through IL-10 TGF-beta, they lose the expression of MHC on dendritic cells. They destroy the expression of co-stimulatory molecules. They decrease the maturation of APCs and their functionality. They decrease the production of inflammatory cytokines. So total immune system is suppressed during the viral infection. So as I told you earlier, that we need to understand in the viral infection that which cells are playing important role, so we can come back and make the biotherapies along these cells. So in summary, regulatory T cells produce TGF-beta and IL-10, which are the suppressive cytokines, and they play a particularly prominent role in curtailing immune-mediated inflammation. However, in viral infections, because they suppress all other cells and IL-10 TGF-beta concentration become more, so these pro-inflammatory cytokines become abundant in the innate immune system. In earlier slides, I explained you how T cells are the real main player in the antiviral immunity, and if these T cells are exhausted. So you will ask me how the T cells will be exhausted. There is one kind of receptor, programmed death molecule, once present on the cells now they can ligand with the APCs, and which are having the PDL1 ligand, and when they both combine PD1 PDL1, then their T cell signaling, downstream signaling is inhibited. Now that downstream signaling does not make any cytokines for proliferation, does not make interferon alpha to clear the virus. However, if the PD1 PDL1 blockage is given, or any kind of biotherapy or antibody against the PD1 or PDL1 is given, now this PDL1 and PD1 interaction is blocked, and the cells will be becoming the functional. They will start secreting the interferon cytokines for the antiviral immunity. Infection exhaust all T cells. Here I am giving you the example of hepatitis B viral infection. And in hepatitis B viral infection, patients who are E antigen positive, they have the PD1, which is the exhaustion molecule present on the T cells. That PD1 expression without the therapy at baseline is significantly more than the PD1 expression after the therapy, after 24 weeks of antiviral therapy. It means the T cells before therapy, these T cells are exhausted, because I explained to you in the earlier slide that PD1 expression is the programmed death receptor expression. If the cells have PD1 expression, those T cells will become exhaust and they are non-functional. In the same way, we have seen in the other picture that the E antigen positive and E antigen negative patients, where the regulatory T cells have the PD1 expression, there was in 24 week that at baseline also the E antigen negative patient had less, but in the regulatory T cells become more. It means the PD1 expression present on regulatory T cells, which increase the PD1, it means regulatory T cells will be lost after the treatment, and immune system will be again in balanced situation. Earlier there was more of regulatory T cells, and which regulatory T cells were suppressing the total immunity during viral infection. However, after the treatment, PD1 expression comes more on the regulatory T cells, and then the regulatory T cells are depleted. Once the regulatory T cells are depleted in 24 weeks, CD8 positive T cells are coming back, and now they are more functional. Here I will discuss how we will make the strategies to enhance the innate immunity, adaptive immunity, and enhance their therapeutic opportunities. How we will enhance the therapeutic opportunities. In the first strategy of the therapeutic opportunity is induction of innate immunity. Boost the immunity, innate immunity through TLRs. TLRs are toll-like receptors. They will sense the viral antigens or total virus. Better they will sense the viral antigens, better there will be the adoptive immunity, which will help to clear the virus. However, if the TLRs or innate immunity is decreased or diminished, so we have to lift up the TLR's expression by giving the agonist to clear that virus. We are here taking the example of again hepatitis B virus, which can be cleared by having injection of TLR 3, 4, 5, 7, 9, which induce the interferon alpha production. In the earlier slide, we explained that how TLRs can be taken as an injection and induce the adoptive immunity. Here, what are the targets for TLR7 and TLR8 to induce the antiviral immunity? Why we have chosen the TLR7 and TLR8? Here I will explain you the toll-like receptors which are present on the antigen presenting cells. They are basically present on the surface. Like there are 14 toll-like receptors. Out of 14 toll-like receptors, TLR3, TLR7, 8 and 9 are present in the nucleus of the cell. To reach the nucleus of the cell, that they recognize the RNA or the DNA viruses. Once you give the induction of TLR7, TLR8 or the agonist for the TLR7, 8, how these cells, they induce which cells? PDCs, plasma cytoid dendritic cells. TLR7 induce B cells, but TLR8 induce the myeloid dendritic cells, monocytes, macrophages and neutrophils. They all together, what they do, they increase the adaptive immunity, they increase the innate immunity, and they have the antiviral cytokines to clear the virus. I will give you here example of one drug company, Gilead company, which has produced the TLR7 agonist for hepatitis B virus induction. They have combined or they have made the agonist named as GS9620, an orally available agonist. Because these TLR7 agonists you can consume orally. But it does, again, as it have the increase the expression or the functionality of TLR7 signaling, it influences the IRF7 and NF-kappa beta signaling. It increase directly, indirectly the interferons which are helpful for the viral clearance and the pro-inflammatory cytokines. But this drug TLR7 is selective for antivirals versus pro-inflammatory response. Preclinical studies shows that it reduces the viral DNA in the animals, especially in chimpanzees and woodchucks. Even in the phase 1 trial of the hepatitis B virus, which is completed, and it shows it is safe in healthy volunteers too. How TLRs, toll-like receptors, totally induce viral immunity? Here this figure depicts that one hepatocyte or the liver cell is infected with the HBV, hepatitis B virus. How these toll-like receptors boost the innate immunity. So left side of figure shows that whenever they see the hepatitis B virus, it goes to the hepatitis B virus genetic material, enter into the nucleus and stay there covalently close circular DNA, which is very difficult to treat, and therefore it's because it stays in the nucleus. However, if you give the TLR or agonist, which are the TLR7 or TLR8, so what happened? It increased the TLR downstream signaling molecule. It boost up the JAK-STAT. It produced the interferon 1 and interferon 3. And indirectly interferon 1 and 3, it inhibits the production of cccDNA. Indirectly, it helps in viral clearance. Now, if you see the right side picture of antigen presenting cell, which is called as the Kupffer cell in the liver, and that you increase the TLR expression, what happened that there is a S antigen molecule which is in the endoplasmic reticulum, and now that this TLR7 expression also increase the IL-12 and IL-18 cytokines, which is also essential for the antiviral immunity. So directly and indirectly, TLRs agonist, they boost up the infected hepatocyte and antigen presenting cells also, because on the antigen presenting cells, once the TLR signaling or TLR expression is high, surface molecule of hepatitis B virus will be cross presented well through MHC molecule to the other T cells. How TLR8 induced inhibition of viral replication? Here we have showed in the picture, once the TLR8 agonist you have used, it enhances the total PBMCs including the dendritic cells. It secretes the particular cytokines, and those cytokines act on the HBV infected hepatocyte. And then you can see the change in the viral parameters. So what the Lopetin et al. Have studied, and they have published this study in 2030 in Antiviral Therapy, they, after the agonist induction with the PBMCs, they have stimulated the PBMCs with TLR8 agonist, saw the cytokines releasing, and in HBV infected hepatocytes, they have observed that when they have not used the TLR8 agonist, the mock experiment, there was more of HBs antigen. However, with the TLR8 agonist, HBs antigen was lowered down. Similar experiment did, and they saw the RNA parameter. HBV RNA was decreased when they have used the TLR8 agonist, because HBV RNA will be cleared more after the TLR8 agonist, which boost the innate and adoptive immunity together. Again, in this slide, I am explaining you the TLR7 agonist induces how antiviral immunity. It increases the CD8 positive and NK cells functionality, and they do secrete the granzyme B more than if you have not used the agonist. Now it increases the functionality of B cells. It increases the immune breaks. It has the expression of more PD-L1 hepatocytes and the Kupffer cells. So TLR7 agonist induces antiviral immunity through monocytes, through NK cells, through Kupffer cells, through CD8 positive T cells. Other than all these cells, how innate immunity will exploit NK cells, monocytes and MDSCs in clearing the virus? Once expanded NK cells, you block its inhibitory properties. So blocking TRAIL-R2 TRAIL concept will enhance the viral clearance. Activation of monocytes as vaccine for personalized medicine can be used, and we can certainly use boosting MDSCs as the biotherapy for the viral clearance. So these were the cells which belong to innate immunity, and we can enhance this innate immunity cells for the viral clearance. Now I will talk about what are the strategies to enhance adoptive immunity and can use these opportunities as a therapeutic opportunities. I'm explaining, as I explained earlier, that there is the PD-1 expression on the T cells. This PD-1 receptor is present on, whenever it present on T cells become exhausted and non-functional. It stops proliferation, it stops cytokine secretion and cytotoxicity. I believe that you understand that PD-1 has the PDL ligand on the antigen presenting cells. So now when PD-1 and PDL, they combine together in a other way, when T cells and APCs, they interact with each other, instead of activating their downstream signaling and producing the interferon or IL-2 or more cytokines, there is no production of these cytokines because PD-1 expression inhibits the downstream signaling. However, now biotherapy has come that where you have the antibody against the PD-1 or antibody against PDL1. Now you block the PD-1 and PDL in between expression which attached with the antibodies, and again T cells become functional. They are not exhausted anymore. They proliferate, they secrete interferon gamma, TNF alpha and IL-2, and they become cytotoxics also. So in a very simple way, blocking the PD-1, PDL1 axis make the T cell functional. In the hepatitis B virus infection, you have to specifically induce or enhance the antiviral cells, CD8 T cell response. So for expansion of this CD8 T cells, I need to block the PD-1 and PDL1 interaction. In vitro studies have shown to the researchers that when you specifically block the HBV viral infected CD8 T cells, and PD-1 and PDL1 is blocked, then there is enhancement of CD8 T cell functionality. However, when you have blocked in the CMV, cytomegalovirus infection, there is not much enhancement of CD8 T cell infection. So researchers have shown that you have to block the PD-1, PDL1 pathway in different viral infections differently. What is the safety parameter with the PD-1-L1 blockage and how it enhances the hepatitis B virus immunity? PD-1 antibody, if you are using, it acts on T cell and it is more efficient blocking. However, it has many adverse events. But when you are using PD-L1 antibody on the APCs, it is less efficient blocking. However, it has less adverse events too. There are three ways to enhance viral clearance or the viral clearing CD8 positive T cells. How? First, you give the antiviral drug, which you start from day 0. Once the antiviral drug is there, you can restore the T cell or the total immunity. At the time of restoration of T cell response, at that time you give the PD-L1 blockade. PD-L1, as I explained, you present on the monocytes. Now this blockade, when you will give, it will give you the more enhancement in the immunity. This is better explained by Michael M. et al. And published. They have these studies in Journal of Hepatology 2014. We have till now read about that how the viral infection and they damage the total immunity and how we can enhance the innate or adoptive immunity of the system. Here I will again give you few hints that how to replace the standard vaccine. There are therapeutic vaccine which we can use in antiviral as antiviral therapy or viral infections. There are DNA vaccines, T-cell vaccines, adenovirus-based vaccine and yeast-based vaccine. Latest is yeast-based vaccine and I will explain you more in detail later on. Now I combined this that how the recent therapeutic approaches have shown that to boost the innate immunity and to boost the adoptive. Upper part I have already explained you that how you can boost or use the TLR agonist and see the boosting of innate immunity. However, down part of this slide certainly shows that you can give the vaccine therapy including the vaccine against HBC, HBS and the adjuvant, and when you give the HBV specific CD8 T cells it enhances the adoptive immunity. You can use the engineered T cells, CAR T cells, you can use the HBV specific T cell receptors and then you can enhance the adoptive immunity. As biotherapy, you can use the even inhibition of exhaustion markers which are PD1 which I explained you. Another exhaustion marker is CTLA4. So antibodies can be used against these to make the survival of viral infection, or there will be these all therapy will act as antiviral immunity. New molecules have done. I am giving the again example of hepatitis B virus infection. So other new molecules are coming, like I will give you the LTBR signaling. To block the or to agonist the LTBR signaling that molecule, because once LTBR signaling is induced by agonist, it inhibits the cccDNA. Another is the CRISPR-Cas9 action which can inhibit the cccDNA. RNA silencing can be used as biotherapy which can use the inhibitor for surface and core antigens. Many other new molecules are coming for antiviral capacity in hepatitis B virus infection. So students, in summary, I will tell you in these modules that we need to use antivirus. However, along with antiviral drugs, biotherapy for the viral diseases is used as TLRs agonist which enhance the innate and adoptive immunity. PD1 blockage therapies can be used, CTLA4 blockage therapies can be used. These both molecules, PD1 and CTLA4, the exhaustion molecules, and if you block these exhaustion molecules, T cell functionality automatically will be better and you need as a biotherapies as antibodies against PD1 and CTLA4. There could be the manipulation of another kind of cells which are called Th17 pro-inflammatory cells. So blockade of these cells may improve the antiviral immunity. Improvised vaccine strategies can also be used, and there are the T cell based adoptive transfer therapies can also be used in this as the to enhance the antiviral immunity in this module. Thank you.